Hi, this is Audrey and Spencer, and we're in the section 7A Beard, and this is our Table B bacteria experiment.
Here's our question. How does the kind of food affect what the average rate of bacteria growth on them when in contact with the ground? We want to know that if you drop food on the ground, it matters what kind of food you drop to make a specific 5-second rule, or something close to it. Here's our hypothesis. If we drop cheese on the ground for the allotted amounts of times, we want to know what the rate of bacteria growth is. We think that it will be moderate because cheese has a lot of lactic acid, which bacteria can use for energy. If we dropped sliced apples after being left on the ground for the time needed, we will find that rate of bacteria growth is high because apples have a lot of water, sugar, and carbohydrates. But we think that bread won't grow a lot of bacteria over time because there aren't many kinds of bacteria that grow on it.
This is our dish setup. We divide the Petri dishes into 4 quadrants, and before we drop a piece of food on the ground, we will take a negative control and inoculate it in the quadrant. After we drop the food on the ground for the given amount of time, we swipe it with a Q-tip and then inoculate it on the second plate. We then parafilm them to avoid contamination.
This is our expected graph. We think that cheese will start with the most bacteria with a negative control because cheese's structure and taste is made up of bacteria. Although we think bread will have the least amount of growth because of its very, very, very specific amount of bacteria that grow on it. We think that apples will have the fastest rate of bacteria growth because of the many attributes that attract bacteria. We need 4 slices from each of our testing foods to lead our bacteria experiment. We need a plate to see the amount of bacteria that the foods attract. We need an incubator for the bacteria to grow. We need Q-tips to inoculate the foods and to swipe on the Petri dishes. And we need parafilm to wrap the dishes so to avoid contamination.
This is our procedure. What you do is you split the plates into quadrants, and then you take a sample of the food before dropping it on the ground and inoculate it in the plate. After you drop a sample of food on the ground, leave it on the ground for the amount of time needed, and then you'll pick it up and inoculate it and swipe it in the plate. After you have inoculated all the plates, cover them in parafilm to avoid contamination and put them in the incubator at 39 degrees Celsius for 5 days. After that, you will take them out and make observations. You will see how much bacteria is on the surface area of the quadrant of the plate. You will determine the rate of growth by the amount of bacteria on the quadrant.
Here are the questions that our group researched. What makes/affects bacteria growth on cheese, apples, and bread? We found all of these attributes that are attached to these foods that attract bacteria. We went to different science sites and learned what the rate of bacteria growth is when dropped on the ground. We made conclusions from this information that we found that will help us determine and conclude our final research data. Here are our data tables. We have data tables for each kind of food: bread, apples, and cheese. We have the average rate of growth per second for each food. For bread, it's 1.82% of the quadrant covered per second left on the ground. For apples, it is 5.58% of the quadrant covered in bacteria when left on the ground. For cheese, it is negative 0.61%. meaning that when you put cheese on the ground, it will purify. Like we predicted, cheese had a high negative control, which we think is because cheese is made up of bacteria. Here's our all food data table. These are the different data points for each food. So after 5 seconds, apples attract the most bacteria, where cheese purifies. After 10 seconds, apples again has the most bacteria. Cheese purifies again. 30 seconds, apples have the most again, and cheese purifies yet again. But for 60 seconds, apples do have the most bacteria, but cheese is right behind it, which is weird because it purifies for the other time.
This is our final graph. We expected in our expected graph, we expected that there would be a steady increase in amount of bacteria per second. Actually, the data points seem to be all over the graph. For bread, there's a steady increase until 10, and then it drops back down. And for apples and cheese, there's an increase, then a decrease, then another increase. Our average happens to be about the same as apples and bread. We think that this may have happened because some of the plates have gotten contaminated, and we think that maybe when we were dropping the food on the ground was different, had different amounts of bacteria on it in the beginning before we started the experiment.
This has been a presentation from Table B Bacteria Experiment.